Hello everybody. Thank you all for staying until Friday and for listening to my presentation. Today I would like to present you a code that we have written, the BOSS code, that helps in cases where data generation in material science is very expensive or very time consuming. This code that we have produced was born out of a review of material science that we have done in the last few years that showed a clear need for this tool. So I will introduce this overview of material science data infrastructures at the beginning of my talk, and then I will tell you more about the BOSS code that we have developed, and then present how it works with two different examples. One is from biomaterial science and the other from surface science. So let's start with a material science overview. So we had a project at Aalto University that involved Lauri Hieman, Amber Gertz, Adam Foster, and myself, where we cataloged the status of data-driven material science and looked at sort of the challenges and perspectives that we could find. And here's what we found. These are all the data infrastructures basically from the 60s until last year, 2019. So at the very beginning in the 60s, there were only a couple, essentially, and it stayed that way until the 90s. And then in the early 2000s, very rapidly, more and more data infrastructures came into being. And the color code here tells you the age, and then the dots in the world map show you the distribution, and you can see that it's very heavily centered on the US and on Europe, and then data structures more recently are also emerging in Asia, like China and Japan. If you integrate over this number and look at the sort of accumulative data infrastructures over the years, you see this very, very steady status quo, where there were essentially just a couple of infrastructures until the mid-90s, and then very suddenly we saw an almost exponential increase, and one of the triggers here was certainly the materials project. So this is an exponential curve that we may not want to flatten, you would want to say, although I expect that at some point in the hype we will also see a phase of consolidation when materials infrastructures will start to merge again into larger conglomerates and entities. Part of this project was then also to look at current challenges, and we grouped them into 5 different categories: those of relevance, completeness, standardization of data, the acceptance of such infrastructures, and the longevity, essentially how long will these structures be around for us to use them. I don't really want to go into the details of all these aspects. You can read about them in the paper, and the reference is given at the bottom. What I would like to highlight, though, are these 2 aspects at the top here of relevance and completeness, and they pertain to the kind of data that is in these infrastructures. Relevance obviously pertains to the kind of the relevance of the data. Essentially, is the data that you want as a user of such an infrastructure in the database. And completeness then addresses the fact, is there enough of the data that you need for your purpose? And oftentimes these days that is not the case. And one of the reasons is that the majority of the data infrastructures that we found that are currently in existence are full of computational data. And this is because computational data is easily attainable and is easily archivable. So what is then in turn missing is experimental data. So how can we incentivize data collection for very time-consuming experiments or very time-consuming calculations, which is also something that is missing from these data infrastructures. One example for such a case of difficult to attain experimental data is found at Aalto University in the Finceres materials cluster. This is a biomaterials bioeconomy cluster where the objective is to harness the natural properties of cellulose and lignin for new materials. Thus far, research approaches in this materials cluster follow sort of a conventional way of materials synthesis and then characterization. But there's a strong need in that community and a strong drive and desire to digitalize data and to digitalize research. However, one of the problems is the aforementioned timescale. Material synthesis can take a long time, sometimes even days per sample. And this means there's not a lot of data sometimes, and every data point becomes quite valuable. So the solution then to gathering such data is to design active learning strategies for optimal data acquisition. A second problem pertaining to this slow acquisition speed is that it then becomes difficult to systematically explore parameter spaces that are given, say, by the synthesis method. Again, machine learning can help here because adding machine learning to the data analysis and data acquisition can then help with inferring data relations and correlations that are present and cannot be explored systematically by systematically scanning the parameter space. So our solution to these two problems is Bayesian optimization, and the tool that we have developed is called BOSS, Bayesian Optimization for Structure Search. You can find the reference here at the bottom. And it's a global optimization code that is based on an active learning algorithm. The code is based on Python, has a very easily usable Python interface, and is freely available under the Apache 2.0 license. And I've included the link here, I'll show it also at the end of the talk, to our GitLab repository where you can go and download the code for free. So let me say a few things now about the BOSS code and how we implemented a smart data acquisition strategy. So what we need, first and foremost, for optimization is an optimization target. And this can be one of many things. So in synthesis, this is often the maximum yield, but it could also be the lowest energy of a structure or a high performance of a feature or a variable, anything you can imagine. So then we generate an initial set of data. This could be synthesis data, could be something else. And to this data, we fit a Gaussian process model. So this is a distribution over functions. And the last thing we need is an acquisition function. So it's a strategy to then determine the next data point at which one should acquire data. And this should be designed such that we acquire with the most benefit. And I explain in the following what that means in practice. So then we add this new data that we have acquired and fit again, and then we repeat this procedure essentially until we achieve convergence, which usually means that we have found the global minimum. So then in our optimization process, we did find the global minimum, but because we have an analytic function by the Gaussian process regression model, we also have the entire landscape and then access to local minima. And this can then be very important because the local minima give us other points of interest, for instance, conformers, and the landscape is essentially the relation that we wish to visualize in complex data if we have just a few data points. As a bonus, basically, this whole process is scale invariant, so the search dimensions can be anything that we want. Okay, a few more technical details. We, as I said, use a Gaussian process, so that is a model over functions. So if we have at the beginning no data, then essentially all functions in a permissible range are possible functions that we can use. And that's our prior distribution. And as soon as we acquire data, we restrict the space for the functions and then demand essentially that all functions have to run through the data points. So in those data points, the variance is then 0. And in between data points, functions can explore a wider range. And then the mean of all the functions is basically the surrogate model that we want. And then the variance gives us the uncertainty that we have in the model in regions where we don't have data points. Then we basically use a kernel, exponential kernel, that has a signal variance that gives us the amplitude of the function, and then it has a length scale that determines the variation of the functions on in the variables. The acquisition strategy that we then devised balances exploitation and exploration, and for that we use the exploratory lower confidence bound. In exploitation, we acquire the next data point very close to the global minimum, and in exploration we venture out into the space and acquire in regions where the uncertainty is high to make sure that no other minimum is hiding there that could be the global minimum. And the acquisition function that we're using is of this type. We have the posterior mean, that's the average of the function, and here's the variance, and we minimize this expression. If we minimize the first term, we get to the global minimum. If we minimize the second term, we minimize uncertainty overall. But this is best explained for a one-dimensional example. So here you see in black the true function of a process that we have pre-computed. And then we have 3 data points, and the average of the Gaussian process is the blue line. And you can see that it runs nicely through the data points, but the current global minimum is not in the right place. And then we have regions here in gray where we have high uncertainty. So the next data point then is acquired in a region of high uncertainty. And it turns out to be quite high, so now we know the global minimum is not there. And the actual prediction of the global minimum is now already much better. And overall, the uncertainty has been minimized too. And then the next data point is exploitation, so we acquire right at the global minimum. And now have a very good global minimum prediction and overall low uncertainty, which we can further minimize by acquiring more data points in that region. And with only 9 data points, we have then completely mapped out the function. So this is very efficient. Okay, let me now give you an example for biomaterials where we apply this strategy. So in this fin-series cluster, one synthesis that was done was to produce oxidized tannic acid nanoparticles. So tannic acid you can get from wood. And if you then add water to this tannic acid and a base, then basically shake the test tube and let the reaction proceed, which takes about 14 hours, you then obtain oxidized tannic acid nanoparticles. So note here, not a very difficult process, but the reaction time is very long, so you don't produce very many samples in your average lab week. There are essentially two control parameters. That is the strength of the base that we add, and then the pH value of the solution. And then we have one objective, that is the dimension of the nanoparticles that are produced. So are they 3D, 2D, or 1D? And this can be measured with scanning electron microscopy. So when you perform the experiments in this pH range and base strength range, then you get one of four outcomes. Either there's no usable precipitate, or there's no quantifiable precipitate where you get some amalgamated growth. In most cases, however, you do get nice particles. So here these rod shapes seen in 3. And occasionally you get a bit of everything. So you get some cubes, so 3D, and a little bit of 2D. So then here are some good examples of successful synthesis. And what is being measured then are the dimensions D1, 2, and 3, and then the range of micrometers. And the average uncertainty on the measurements is about 1.3 micrometers. So what we then do in the BOSS procedure is we normalize all the data input to dimension 1 equal to 1. And then our objective function for quantifying the dimensionality is D2 plus D3. And we take the negative. So then the global minimum would be a 3D structure, and the maximum of that function would be a 1D structure. And then we introduce noise, which is no problem in this Gaussian process, to account for the experimental uncertainty. So what we then get with only 10 data points in this Gaussian process is this intuitive landscape where you have a 3D minimum here in this pK, in this base range and this pH. And then the 1D region would be up here. And then we acquire 7 more data points, so 7 more synthesis points were produced. And what we find that this was actually quite important, because now we're probing this region that had a very high uncertainty before because it had no data. And now actually the 3D region shifts to the bottom left of the diagram, and the 1D region becomes more refined up at the top. So we see a clear trend in this variable space that we go from a 1D growth to a 3D growth as we decrease pH and the base strength. So with only 17 data points in this 2D optimization space, we can then produce a very intuitive map that characterizes this process, where we have essentially a 3D growth region, a 2D growth region, and a 1D growth region. And then in between, we have these transition regions where other stuff happens. And if we now double check with the micrographs and put them basically in at the right points, we see indeed in the 1D region we have these rod-like nanoparticles. In the 3D region we have nice little cubes down here. In the 2D region we don't have too many samples, but you can see we get these nice little platelets here. And then in these intermediate regions you essentially get something that is pretty much intermediate. You get these little stubby 1D, almost 2D-like structures here and here. Also in this intermediate region between 2D and 3D, we get something that is between 2D and 3D. And then we have some images that the experimentalists said they're non-quantifiable, and they also fit nicely into these intermediate regions. So to sum up this part, the BOSS approach helped us to produce experimental data in regions of high interest. It then also produced intuitive correlation maps that gave us insight into the process. So we are very data efficient, but it is still, as we found out, actually often hard to convince experimentalists to pursue such an active learning approach, because they're very reluctant to go back to the lab over and over again to do more and more experiments, which is something they would need to do. Okay, the last example comes from surface science. So we teamed up with colleagues that are doing atomic force microscopy, Ben Aldred and Peter Liljeroth, and they imaged a 3-dimensional molecule on the copper surface, this camphor molecule. And this is a very challenging task in AFM experiments. And you can see this here. So these are micrographs. They look different, so there's an indication that they're different structures of the camphor, but you don't really see very much in these images. So the solution then is to perform electronic structure theory calculations with density functional theory for this system, and then get atomistic insight, simulate AFM images, and compare to find good matches. The problem with this is these systems are large, 219 atoms, so the DFT calculations require a significant computational budget for each, and in addition we would have to explore many configurations to sample this configurational space of the molecule, so this would be extra expensive if we followed a conventional structure search approach. So again, we use the smart sampling that our BOSS tool offers to reduce the number of calculations and only do those that carry the most information. So here I'm showing you a video of how the BOSS approach works. So on the left you see basically the structures that BOSS explores, and on the right you see the current global minimum. So BOSS basically moves the molecule wildly through space, and this is essentially all the exploration that we do, whereas the global minimum prediction is converged very quickly in its final form. So in total we need about 900 single point DFT calculations, so no geometry optimization, and that it amounts to approximately 50% of what a conventional structure search approach would take. So we are quite efficient. So then to sum this up, we then obtain 8 different structures. So here I'm showing you the first 5 that have the lowest energy, and those have been now extracted from the potential energy surface in the local minima. So this is the extra information that we get from having the full functional form. You can see the lowest energy structures all have oxygen pointing down to the surface, so there's a chemical bond. And then we find 3 others that have a lower binding energy, and they all have the oxygen pointing up, so it's physisorption. And then we did the comparison. We simulated AFM images for these structures, compared them to the real AFM images, and what you can see then is that the match is quite good actually. So we were able to identify 3 conformers of this molecule, 3 different orientations of this molecule on the surface. So we have some kind of polymorphism for this molecule. Okay, so the conclusion of this part is that for the first time we were able to identify different geometries of a 3D molecule on a metal surface, and were able to unambiguously assign the structure to the AFM images. And again, BOSS made this very efficient. Okay, I'd like to conclude then this talk with a quick summary of our BOSS code. Again, here's the URL for you to download it. So we have a robust implementation of the GPy Gaussian process with an easy input file. There are restart capabilities so that you can use it on HPC platforms. There's extensive post-processing, extensive visualization, easy to use Python interfaces. There are various kernels and acquisition functions that we have implemented that you can choose from. There are priors on hyperparameters you can use, automate a minima extraction, and many other tools. One thing that should now be added to BOSS is a capability or a feature that interfaces to existing data infrastructures, so that data that was recorded with the BOSS code can then automatically be uploaded to platforms like the Materials project or Nomad, for instance. Finally, I'd like to show you the BOSS developers team. The real driver behind BOSS is Milica Todorovic, who together with Yuka Koranda and Mikhail Gutman conceived the initial idea, and she's now leading the development that is done, for instance, by student Jari Järvi, Henri Paulamäki, and Ville Parkinen. With this, I've come to the end of my presentation, and I'd like to thank you for your attention today, and I'm open for questions. Thanks a lot.